Biotechnology and its applications. Introduction. Biotechnology uses living organisms or their components to develop useful products and technologies. Like biopharmaceuticals and biological using genetically modified microbes, fungi, plants, like that. And the application of biotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. And means three critical researchers present, providing the best catalyst in the form of improved organism, or creating optimal condition through engineering, downstream processing technologies. These are next one is biotechnological applications in agriculture. In that green revolution, use of improved crop varieties, better management practices, and agrochemicals like fertilizer and pesticides increased yield, but it was costly. Means green revolution succeeded in tripling the food supply, but it was not enough to feed the growing human population. Increased yields have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices, agrochemicals, but it was very costly. Next one is tissue culture, growing plants in sterile nutrition medium and based on tortipotency. Tortipotency means the capacity of the cell to produce whole plant from a single cell or any part of the cell. We call it as tortipotency, or any part of the plant means we call it as explant. In 1950, the whole plant could be regenerated from explants. Explant means any part of the plant taken out and grown in a test tube under sterile condition. Uh, and micropropagation. Large number of identical plants, the plants are produced in short duration. This one is called micropropagation. Means a large number of plants are produce thousands of plants through tissue culture is called micropropagation. Each of these plants will genetically identical to the original plant from which they grown. These are called as a summa clones. And meristem culture means if any plant infected with a virus, but the meristem, apical and axillary is free of virus. Hence, we can remove the meristem and grow it in vitro to obtain virus-free plants. And somatic hybridization. Somatic hybridization means fusion of protoplast. Protoplast means cell without cell wall from different plants to get new hybrids. Uh, means isolated protoplast from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplast, which can be further grown to form a new plant. These hybrids are called as somatic hybrids. Mm. This process is called as somatic hybridization. Uh, in tortipotency, we should clear that nutrition medium should contain or provide a carbon source such as sucrose, and also inorganic salts, vitamins, and amino acids, and growth regulators like auxins and cytokines. Okay, somatic hybridization means the fusion of protoplast from two different plants to get new hybrid. Meristem culture means we have, if a plant is infected by any virus, then meristem, apical or axillary is free of virus. Then we can remove the meristem and grow it in vitro, and we can produce a plant. And the example of somatic hybridization means pomato. Pomato means hybrid plants combining of two, tomato and potato. Like these are the example of somatic hybridization. And next one is genetically modified crops. Genes are altered to obtain desirable traits. Plants, bacteria, and fungi and animal whose genes have been altered by manipulation are called as genetically modified organism. There are uses are there genetic modification uses. These are made crops more tolerant to abiotic stresses like cold, drought, salt, and heat, and reduce reliance on chemical pesticide like pest resistant crops, and help to reduce post harvest losses and increase the efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil, enhanced nutrition value of food. Example, golden rice. Golden rice is rich in vitamin A and enriched rice. Means genetically modified crops advantages means these are tolerant to abiotic stresses, means they are they can survive in abiotic stress like a cold, salt like that, and resistance to pest and diseases, and reduces post harvest losses and efficient use of nutrition. And genetically modified plants are also used for industrial raw material, starch, biofuels, and pharmaceuticals, and other applications in agriculture. Production of pest resistance and disease resistance plants, and biofertilizers, rhizobium, azobacter, and biopesticide means Bt toxin. And industrial applications means microorganism used for large scale production of enzyme, organic acid, alcohol, antibiotics, and fermentation of fermentation used in curd, bread, and cheese, vinegar. Bioreactors used for control production and single cell protein like that. And environmental applications means bioremediation, microbes degrade pollutants and toxic substances, sewage treatment using microorganisms, bioleaching, extraction of metals from ores. And next one is bt cotton. Bt cotton means bacillus thuringiensis. It is a bacteria. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide assistance to insects without need of fertilizers, sorry, without need of insecticides. uh, Bt cotton contains a gene, cry gene from Bacillus thuringiensis. This gene produces Bt toxin insecticidal protein, which is toxic to bollworms. Toxic exist as inactive protoxins converted into active toxin in alkaline gut of insect. Active toxins bind the gut cell, pore formation is occur, and cell lysis will occur after the death of the insect will occur. Means some strains of Bacillus thuringiensis produce proteins that kill certain insects such as Lepidopterans, means tobacco budworm, armyworm, and coleopterans, beetles, and dipterans, flies, and mosquitoes. Bee thuringiensis form protein clusters during a particular phase of their growth phase. It is very important to note it is a particular phase growth. Bt thuringiensis forms protein crystal during a particular phase growth, not different phase growth. And these crystals contain a toxic called insecticidal protein. Actually, beta toxin protein exists as inactive protoxin, but once an insect ingests the inactive toxin, means it is converted into active form of toxin due to the alkaline pH of the gut, because it's soluble as the crystals. And the activated toxin binds to the surface of the midgut epithelial cells and creates some force and cause cell swelling, and lysis will occur, and eventually death will occur of the insect. And next one is bt toxin gene. Cry genes. Example, cry 1ac, cry 2ab, cry 1ab. From b from b and inserted into crop plants to make them insect resistant. Each gene is specific to a target pest. Cry1AC and Cry2AB for cotton bollworms, and Cry1AB for corn borer. It means specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crops, plants such as cotton. The genes depends upon the crop and the targeted pest, as most BT toxins are insect group specific. The toxin is coded uh, Cry1AB for corn borer. Means the toxin is coded by gene Cry1AC named Cry. There are a number of them. For example, the protein encoded by the genes Cry1AC and Cry2AB control the cotton bollworms. That of Cry1AB controls corn borer. And the figure represent A destroyed by bollworms and B a fully mature cotton ball. And next one is pest resistant plants. Nematode Meloidigina incognitia infects the root of tobacco and reduces the yield. RNA interference is used to silence specific mRNA of nematode. Double-stranded RNA binds to the mRNA and prevents its translation. Using agrobacterium vectors, double-stranded DNA RNA is introduced into plants. Transgenic plants expressing double-stranded RNA become resistant to the parasite. Means nematode parasites as a nematode Meloidigina incognitia infects the root of tobacco plant and causes a great reduction in yield. A novel strategy was adapted to prevent this infestation, which was based on the process of RNA interference. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of specific mRNA due to the complementary double-stranded RNA molecule that binds to and prevents translation of mRNA silencing. Means if translation have to occur means the RNA should be the single stranded, but in this the double stranded RNA will be forming. In this reaction, in this double stranded RNA, the translation will not be occur, so it is called as a silencing process. Means the nematode RNA is bind by a infectious virus, RNA genome or mobile genetic elements or transposons that replicate via an RNA intermediate. And using agrobacterium vectors, nematode specific genes were introduced into the host plant. The introduction of DNA was such that it produces both sense and antisense RNA. Sense and antisense means these both are complementary to each other, and these both form a double strand in the host cell. These two RNAs being complementary to each other form a double stranded RNA that initiate RNAi and thus silence the specific mRNA of the nematode. The consequences was based that the parasite could not survive in this transgenic host. So transgenic plant therefore got itself protected from the parasite. Next one is biotechnological applications in medicine. In the insulin gene from human is inserted into E. coli or yeast. Bacteria or yeast produce human means the recombinant DNA technological processes have made the immense impact in the area of healthcare. Recombinant therapeutics do not induce unwanted immunological responses. Um, At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed in India. And next one is insulin. Insulin is used for diabetes. We know that. And these are earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source the caused some patients to develop allergy or other type of reactions to the foreign protein. And insulin consists of two short polypeptide chains, chain A and chain B, and these are linked together by disulfide bridges. Uh, In mammals, including humans, insulin is synthesized as a prohormone. Prohormone means it is an inactivate form. Inactivate form means it contains C peptide chain also. This C peptide chain is not present in the mature insulin and is removed during the maturation into insulin. The main challenge for production of this insulin is rDNA technology. And in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding A and B chains of human insulin and introduced them in plasmids of E. coli. Human gene isolation of gene after it is transferred to a plasmid and recombinant plasmid, then it is inserted into E. coli or yeast, then with production of purification of human insulin. Next one is gene therapy. Gene therapy aims to treat genetic disorders by introducing a normal gene into the patient's cells. A normal gene is inserted into a vector, usually by using a virus as a vector, and transferred into the target cells. The modified cells express the normal protein and help in correcting the gene defect. Means gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows correction of gene defect that has been diagnosed in a child or embryo. Here, genes are inserted into person cells and tissues to treat a disease. Correction of a genetic defect involves delivery of normal genes into the individuals or embryo to take over the functions of compensate. Uh, the first clinical gene therapy was given in 1990 to a 4 years old girl with adenosine deaminase deficiency. This enzyme is crucial for the immune system to function, and this enzyme is crucial for immune system to function. And in some children, ADA deficiency can be cured by bone marrow transplantation, and in other it can be treated by enzyme replacement therapy, in which the functional ADA is given to the patient by injection. It can be two types: somatic cell gene therapy and germline gene therapy. Target somatic cells or body cells effects are limited to the individual. Germline gene therapy means target gametes or embryos. Effects are heritable but not yet accepted ethically. Means in some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But the problem with both of these approaches, these are not completely curative. So the patient requires periodic infusion of such genetic engineered lymphocytes. The gene isolate from bone marrow cells producing ADA is introducing to cells at early embryonic stages. It could be a permanent cure for that. Next one is application: SCSI, cystic fibrosis, hemophilia, cancer, etc. It is very important to note the bone marrow transplantation is the permanent cure of the ADA deficiency disease. And next one is molecular diagnosis. Molecular diagnosis is to detect the disease at the genetic level using DNA or RNA or their products. It is more accurate, sensitive, and early than conventional methods. Conventional methods means serum and urine analysis, etc., but it is not early detection. Means it will not identify when the pathogen is just entered into a body. When the pathogen will start to show the symptoms of the disease, then then only it can identify the disease. Further techniques means DNA or RNA probes, PCR, ELISA, gel electrophoresis, and DNA sequencing, and RFLP. A single stranded DNA or RNA tagged with a radioactive molecule called probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will have hence not appear on the photographic film because the probe will not have complementary with the mutated gene. ELISA is based on the principle of antigen antibody interaction, means when the infection when the in, when the infection by pathogen can be detected by the presence of antigen, means when the pathogen infects the body, then only the antibodies are synthesized against the pathogen. So when presence of antigens like proteins, glycoproteins, or by detecting the antibodies synthesized against the pathogen, then only the anti it is the principle of the ELISA. Next one is transgenic animals. Animals in which foreign DNA is inserted in to express extra genes are called as transgenic animals. And example transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced. 95% of all existing transgenic animals are mice only. And normal physiology and development, used to study regulation of genes and their effects on body functions and development, helps in understanding complex biological process. Means transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its developments. Example study of complex factors involved in growth such as insulin like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects. And study of diseases, used as models to study human diseases and helps in developing and testing new treatments. Means many transgenic animals are designed to increase our understanding how genes are contribute to develop of a disease, and these are specially made to serve as models for human disease so that investigation of new treatments for diseases is made possible. Example like such as cancer, cystic fibrosis, rheumatoid arthritis. We know that rheumatoid arthritis is a autoimmune disease, like that. Next one is biological products. Produce useful protein in their milk or tissues. Example human alpha 1 antitrypsin. It is used to treat emphysema. Proteins for PKU, means phenylketonuria and cystic fibrosis. First transgenic cow is Rosie, produced human protein rich milk. Means medicines are required to treat certain human disease can contain biological products. Both such products are often expensive to make. Transgenic animals that produce useful biological products can be created by introduction of the portion of DNA. Means transgenic animals that produce useful biological products, it can create by introduction of portion of DNA which codes for a particular product, such as human protein alpha 1 antitrypsin. It is a enzyme used to treat emphysema. And similar attempts are being made for a treatment of phenylketonuria and cystic fibrosis. And in 1997, the first transgenic cow, Rosie, produced human protein rich milk. It is in 2.4 grams per liter. The milk contained human alpha lactalbumin. It is important to note that human protein rich means alpha 1 antitrypsin, and the milk contained human alpha lactalbumin and was nutritionally a more balanced product for human babies. And vaccine safety, used to test safety and efficacy of vaccines before human use, helps in replacing animals like monkeys in some tests. Means transgenic mice are being developed for use of testing the safety of vaccines before they use it to humans. Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Means the if any vaccine is discovered, it first test on experimentally test on rats only. If then successful, then it will be experiment or it will be used in humans. Next one is chemical safety testing. Used to test toxicity of drugs and chemicals more sensitive to toxic substance result in less time. Means toxicity or safety testing, this procedure is same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substance than non-transgenic animals. They are then exposed to toxic substance and effects studied. Toxicity testing in such animals will allow us to obtain result in less time. Mm, And next one is ethical issues. Genetic engineering raises ethical, social, and moral concerns. Possible misuse and risk to environment and future generations. Unpredictable effects when such organisms are released in ecosystem. Need of laws and guidelines for safe and responsible use. In India, Genetic Engineering Approval Committee regulates GM research and use of GM organism. GM is genetically modified. Plants and genetic materials, plants and biological resources may cause public anger. Means the manipulation of living organism by human race cannot go any further without regulation because some ethical standards are required to evaluate the morality. Therefore, the Indian government has set up organizations such as Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and safety of introducing medicine sources, has also created problems with patents granted for the same. And there is a growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic materials, plants, and other biological resources that have long been identified, developed, and used by farmers and indigenous people of a specific region or country. And next one is importance of biodiversity. Biodiversity is the presence of a wide variety of living organisms in a given area. In India, about 2 lakh species, about 8% of the total global biodiversity is safe. It is our moral, ethical, and constitutional responsibility to preserve biodiversity. It is important. Rice is an important food grain, the presence of which goes back thousands of years in Asia agriculture history. There are estimated 2 lakh thousand varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct for unique aroma and flavor, and 27 documented varieties of basmatis are grown in India. There is a reference to basmati in ancient texts, folklore, and poetry. In 1997, an American trademark office this allowed the company to sell a new variety of basmati. In the US and abroad, this new variety of basmati had actually been derived from Indian farmers' varieties. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. But it is a Indian product. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Okay. Next one is conservation of biodiversity. Protection of species in their natural habitat, example national parks, sanctuaries, biosphere reserves, and protection outside natural habitat, example zoological park, botanical gardens, seed banks, gene banks, like that. Biodiversity acts as a life support system and is essential for human survival. Mm. And next one is patent related issues. Patent system encourages innovation by providing financial benefits. It may also lead to misuse exploitation of biological resources and by and eh, and indigent exploitation of poor and indigenous people. Need for laws to ensure fair and equitable sharing. Next one is biopiracy. Misuse of biological resource by multinational companies without permission from the country or people concerned results in economical loss and exploitation of local communities. India has taken steps to protect biodiversity through laws and international agreements. And most of the industrialized national nations are rich financially but poor in the biodiversity and traditional knowledge. In contrast, developing and underdeveloped with a rich biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop the modern applications and can also be used to save time. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure during their commercialization. There has been growing realization of injustice and inadequate compensation and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patents Bill that takes such issues into consideration, including patent terms, emergency and research and development initiative.